Good afternoon, everyone. I'm Kayal, so I will be giving the talk first, and then Swetha will take over. I'm a grad student with the Aguilar Lab at Purdue University. So first, before I go ahead with the project, I just want to tell you how we came about doing this project, and thanks to the Center for Science of Information for giving us this opportunity. So the project started with the summer workshop, which was held in 2014 at Purdue by the Science of Information. And this was for Science of Information on biological data, specifically for analysis of biological data. And the organizers were Dr. Claudio Aguilar from the Department of Biological Sciences at Purdue and Dr. Mark Ward from the Department of Statistics at Purdue. So they basically brought together biologists and other students interested in studying biology but had background in information theory, programming, and mathematics. And these were students from the different universities which came under the Center for Science of Information. So this workshop happened over an entire week, and we had a very good conversation between the two distinct groups of biologists and the other group who wanted to do biology but had a more background on programming. So basically the biologists brought a lot of data which had to be analyzed, and the other students helped us come up with automated modes for quantifying our data. After the 1 week of interaction, we realized what were the missing bridges between the two groups, and now we were able to communicate better if both the teams were put together. So this led to an interdisciplinary research grant that we were awarded from the Center for Science of Information. And this essentially had 5 students, so this was entirely a student-based team. And the Center for Science of Information encouraged us to apply for this following the summer school, and we also were awarded this team. So me and Swetha from Biology Purdue were a part of it, and it also has Yukon from Purdue itself, who is also a grad student, and Priya, a grad student from Texas A&M, who's from Electrical and Computational. And finally, postdoc Sundar, who is from Civil Engineering and from Johns Hopkins University. So now I'll tell you about the project that me and Sundar and Yukon worked on. So today I'm going to be talking about quantitative analysis of endocytic sites. So I will give you a little bit of background about endocytic sites and the biological relevance of studying endocytic sites. So cancer, how is cancer and cell migration related? Cancer is basically an uncontrolled growth of cells which results in an abnormal formation or abnormal mass of cells. So nowadays it's become relatively easy to control a primary tumor which is localized to one specific region. However, when the cells of the tumor become migratory and travel to distant sites and set up base somewhere else, which is called metastasis, also the spread of cancer, the situation becomes more complicated. So while comparing cancer and cell survival, when the cancer is metastatic free, the rate of curing cancer is almost 90%. However, when metastasis is involved, the cure rate falls down drastically to just 20%. And the difference between the two types is basically the ability of the tumor cells to now migrate, which is why studying cell migration in tumor cells is extremely important. So how is cell migration and endocytosis involved? So this is a schematic representation of a cell with its nucleus and cytoplasmic material. So the cell is usually attached to the surface with the help of adhesion receptors, which are represented here in red. So how does a cell move from point A to point B? When the direction of migration is established, the cell usually puts out a protrusion or an arm, and that's when our star protein comes into play. So the protein that we are interested in studying is epsin, which is represented here as a star. So this comes in contact with the adhesion receptors and incorporates it into these vesicular structures, which then get shuttled to the front end of the cell. So this continuously happens until the cell eventually moves to point B. So just a zoom in of the area where our favorite protein or the protein that we're interested in studying is situated. So this is epsin, and it gets associated with the adhesion receptors, and they are basically internalized, taken into the cell, which is called endocytosis. And epsin, why are we specifically looking at epsin in endocytosis, is because epsin is specifically found to be overexpressed in migratory cancer cells. And additionally, epsin has been abnormally found to be overexpressed in other invasive cancers, which means they are more metastatic cancers such as pancreatic, breast, and skin cancers. I just spoke to you about endocytic sites, which are found at the bottom of the cell, but there are other endocytic sites also present in a cell. So there are endocytic sites present on the top of the cell as well, which are represented here, and their function is just to bind to nutrients in the media and internalize them. So epsin is involved in both the types of endocytic sites. So if we were to take a section of the cell at two different regions and look at them, so this is the cross-sectional area seen from above. So this represents the endocytic sites on the top of the cell, and this represents the endocytic sites at the bottom of the cell. And as you can see, they are different in size, and that is actually true. So the ones on top are smaller and ones at the bottom are larger. So the ones on top are called pits, which play a role in cell eating, whereas the ones at the bottom are called plaques, and they are known to play a role in migration. So this is a picture showing the pits and the plaques as seen by a microscope. So this is each of these dots that you see represents this star protein that I'm talking about, epsin, and that has been tagged with a fluorescent probe. So now you will be able to visualize it inside the cell. So these are pits which are much smaller and fainter on the top of the cell, whereas you can see that the plaques are more brighter and bigger. So we are basically interested in studying epsin and how it contributes to cell migration. So what we did is we have the wild type protein, which is the star protein, and additionally we made different mutations in this star protein by deleting different regions of these star proteins. So these are just representative mutants, let's say. And from comparing the wild type protein to these mutants, which lack different regions, we were able to identify certain mutants which were defective for migration. So we know that the missing regions are the one which are required for migration of the cell. So now that we identified these mutants, we want to know how these different mutants affect the endocytic pits versus the endocytic plaques. And what are the specific characteristics that we want to study? So we want to know if the number of the pits or plaques are affected, or the area and the size, or just the intensity, which is the brightness of the pits or plaques, and finally the dynamics of the pits or plaques. So I'm not showing you movies today, but these pits are much faster, and whereas the plaques are much more stable in comparison to the pits. So these are all characteristics that we want to study. So as you can see, I have one wild type versus 18 different mutants that I want to do this analysis on. And doing this manually, and just one cell has loads of dots. And when we want to study biological data, we need a lot of replicates. So that would mean counting lots of dots, at least thousands of dots, and in hundreds of cells. So having an automated method to do this would be really helpful. So the major challenge that we faced with respect to this project was that already present softwares had a problem with particle detection. They were not doing it very accurately. So this is a picture which shows only one tiny part of the cell, which has a lot of dots. So if I were to manually count this, I get this number, 126. But if I apply the existing software, the number of particles appears as 32. This is because the software detects boundaries of the dots, and when two dots lie very close together and there is some noise in between them, they are considered as just one dot. So we needed a better algorithm to detect particles. So we came up with a strategy during the summer workshop of 2014. And firstly, it was to apply a threshold to eliminate the background noise, and then find the local maxima of dots. Rather than going for boundary detection, we wanted to look at the local maxima. And then the next step was to look at the neighborhood pixels of the local maxima and make sure that they have a non-increasing value, which would mean we would now be able to tell two different dots from each other, even if they lie really close. So this is example of a picture which has our protein epsin tagged with a fluorescent probe. And when we applied the algorithm on it, this is the result that we get. So you see that the local maxima are all picked up, and it was pretty close to the manual counting number that we got. Then followed by this, now we wanted to determine the area of these dots, and I'll just describe the strategy that we came up for this, and this was developed by Dr. Sundar from Johns Hopkins. So as you can see, so these are the pixels, which all the pixels that are shown here are above the initial threshold that we gave the program, and the ones that are highlighted in color are the local maxima. So now the second step, once we detect the local maxima, is now to go in and measure the distance of each pixel from each local maxima. For example, so we start with this pixel and measure its distance from different local maximas. And when you consider this pixel, it is closest to D2, or D2 is the shortest distance, and hence this pixel gets associated with this local maxima. So doing the same thing progressively, it basically populates the field with where these pixels are associated. So finally, the entire image would look like this. So this is a local maxima, and all these dots belong to this specific local maxima. So this would be a cluster, this would be a cluster, and this would be a cluster. And all the intervening dots fell in the background threshold. So how would this look on an image? So this is the cell image with lots of dots, and you can see that they are clustered. And even if they are close together, they are able to be shown as two different dots rather than being considered as one particle. But however, we saw a problem here. So you can see some orphan dots being detected. And what these orphan dots are basically, they were detected as local maxima because they were above the threshold, but then they were too tiny to be a particle because they do not have enough pixels surrounding it. So basically, we need to get rid of them. Otherwise, they get associated with some other clusters, and definitely that is not right because there is a huge distance between them and the cluster. So further improving the algorithm, so in an entire bigger picture, you can see that there are some random dots here which do not have a local maxima. So further improving the algorithm, we were able to get rid of the orphan dots, and now you only have local maxima and its associated cluster. So the entire picture has been cleaned up now and the orphan dots being eliminated. But we still have some problems that need to be solved. Like, for example, me as a user can definitely detect dots over here, which have not been picked up by the program. And the only explanation we have is basically these dots fell behind the threshold that the user inputted. So still there is an user interface happening here. So to eliminate the fact that the user has to input a threshold, we are now thinking of a strategy where the program can determine its own threshold. So here we have a plot which shows the number of particles that are detected as a function of the threshold. And what we want to do is, depending on this graph, if the program itself by iteration can determine a perfect threshold where the number of particles are more accurate. So we are still working on incorporating this part of the code with the previous detection and the area detection algorithms as well. So in future, we want to incorporate the automatic threshold determination. And additionally, the program that we've developed can be used for determination of percentage overlap between two different markers inside the cell. So let me show you an example of that. So this is a cell which has two different components. So one which is marked in green, it is the star protein, which is its original form, and it's tagged with a green fluorescent protein. And then we have another marker inside the cell which has a red tag on it. And what this does is it marks all the endocytic sites that I was talking about inside the cell. And here you can see a merged image where both the red and the greens come together, and wherever they overlap you can see a yellow color. So for example, with the wild type, you can see that almost 100% of the red dots are occupied by the green dots. However, if we go in and make mutations, so what do these mutant proteins now look like? So now this mutant protein has been expressed in a different cell, and then you have the same cell which has our marker and shows all the endocytic sites. So in the merged image, we can clearly see that there are more red dots which are not occupied by green. So it would be really useful to be able to detect the percentage of co-localization between the red and the green, that is the number of red dots that are also occupied by green. And that would tell us the ability of these mutants to localize at endocytic sites, which will very naively translate to their ability to also enhance cell migration. So was that clear? Okay. So conclusion, so till now, we have a strategy for particle detection, a strategy for area determination of these particles, and then we need to make improvements in terms of automatic threshold determination. And finally, we would also like to apply this to determine co-localization between two proteins within the same cell. Thank you very much. And if you have any questions, please reserve them for the end. And the next speaker is Swetha Ramadesikan.
A very good afternoon to all those present at this meeting. I'm Swetha. I'm a second-year graduate student in the Department of Biology. I work in Dr. Aguilar's lab, and today I'm going to be talking about the role of OCRL1 in Golgi-related defects in Lowe syndrome. So that is the project that I'm working on, and very briefly I'm going to be introducing the biological aspects of the project, and I will also be talking a little bit about what we did at the summer workshop that the center organized, and where we are now with respect to developing a tool to quantitatively analyze the biological data that I'm working with. So, very briefly, what is Lowe syndrome? It's a rare genetic disorder that was identified in the 1950s, and it was later identified that mutations in the gene OCRL1 is known to cause Lowe syndrome, and mostly males are affected by this disease. So, male children who are born with Lowe syndrome, they have cataracts in their eyes at the time of birth, and by 1 year of age, they develop kidney failure and other kinds of renal problems, and also exhibit mental retardation and cognitive delays. So over the years, the kidney dysfunction just worsens over time, and eventually these patients succumb to end-stage renal disease, which ultimately kills them by the second decade of their life. So far, there has been no known cure for this lethal disease, and our knowledge about how mutations in a particular gene leads to this disease is also very limited. So at this point, what the field does know is if a cell loses OCRL1, which encodes a protein that goes by the same name, OCRL1, or if a cell is deficient in this protein OCRL1, there are a lot of cellular defects that these cells exhibit, which incidentally our lab was the first to identify in the Lowe syndrome field. So our lab identified many novel cellular defects associated with the disease. But what we still do not know is how do these cellular defects ultimately lead to the disease that we see in these patients. While there are many defects in these patient cells, and these defects affect many parts of the cell, the one part of the cell that I'm very interested in and I'm going to be focusing my talk on is what is called the Golgi apparatus. So the cell can be thought of like a big machine that has multiple parts in it, and all these parts have to be working together in sync in order for the cell to function correctly and perform all its roles in the larger picture, such as in an organ or in the whole human body. So one such part of the cell that I'm going to be talking about is called the Golgi apparatus, and in the cellular or in the biological context, these parts of the cell are called organelles. So the Golgi apparatus, or the Golgi as it's shortly known, is an important or the central organelle in the secretory pathway. So in very simplistic terms, the Golgi can be thought of as the USPS of a cell. So the cell produces vast amounts of proteins, vast amounts of lipids, and all such things. And all the things that are produced by the cell need to be sent to a central location, which is the Golgi in this case. And from there on, the Golgi modifies all these components, associates signals or tags on them, and ensures that they're all sent to different parts of the cell and at a given time or with a given signal. So as I mentioned, the Golgi apparatus is very, very crucial for sending various proteins and lipids to various parts of the cell. And incidentally, the protein that I'm interested in, OCRL1, which I would like to remind you is deficient or completely lost in patients with Lowe syndrome, is located in the Golgi, as you can see here. So this is a pictorial representation of the Golgi, and the green particles here represent the protein of interest, OCRL1. So I just said that we have already identified many cellular defects that are produced when a cell loses OCRL1. And given that it localizes to the Golgi, I'm interested in understanding what are the defects that the Golgi apparatus would be faced with when a cell loses OCRL1. So on the leftmost panel here is a fluorescence image of a normal or a healthy cell. So the Golgi apparatus is fluorescently tagged with a red fluorescent probe. So it lights up only the Golgi, whereas the nucleus of the cell is tagged with a blue color probe, so that lights up the nucleus alone in blue. So on the left panel, you see normal cells that are stained for the Golgi and for the nucleus, and you can observe that it's very compact and very close to the nucleus. However, the middle and the right panel that we have here are cells from two different Lowe syndrome patients, And as you can see, the Golgi in these patient cells is highly dispersed. So it occupies a larger area in the cell when compared to the normal cell. And in some cases, the Golgi is also very discontinuous. So in normal cells, it's going to be a continuously packed stack of ribbons, if you can imagine that, whereas in patient cells, it looks like it's discontinuous and dispersed at the same time. So visually, we are able to see that they are very different. But as cell biologists, we are not only interested in qualitative information, but we're also very interested in trying to understand how quantitatively these biological phenomena are different in order to provide a statistically significant biological result. So the objective of my study is to quantify the differences in Golgi structure in cells from healthy volunteers and compare them against cells from Lowe syndrome patients. And as I mentioned, the Golgi is very important for trafficking various proteins and lipids to different parts of the cell. So it is a very dynamic organelle, and we would also like to see how the dynamics of this organelle changes in disease and in normal conditions. And finally, in this disease, there have been over 100 mutations that have been identified, and all these mutations affect the same gene OCRL1, and different patients carry different mutations and still display symptoms of Lowe syndrome. So we want to identify how the Golgi changes across these different patients who have different mutations in the same gene. So the next question is, why would this information be relevant to me and for the scientific community? So by able to quantitatively assess changes in Golgi morphology in these patients, we would be able to stratify them or categorize them into different groups as to, depending on whether the Golgi abnormality is very severe or very mild or intermediate. And this kind of stratification would provide us clues towards correlating the severity of disease with the behavior of the Golgi. And a very important aspect of my project also deals with identifying therapeutic strategies that are specific to Lowe syndrome. So I would like to remind you that this disease, despite its identification in 1950, there is still very little known about it, and there is still no specific cure towards the disease. So by developing a quantitative method to determine morphological changes in the Golgi, it can be used as a very good readout to study the effect of candidate drugs in their ability to rescue this particular cellular defect that I'm working on. And finally, it is going to be a very, very objective method of analysis, and it wouldn't be subject to the bias of the user or someone who's performing the analysis. So while I started trying to quantify these changes in Golgi morphology and structure, some of the biggest challenges that were associated with this kind of analysis was I was dealing with a large amount of data. So we are talking in terms of easily 50 images, and each image would have about 10 cells. So we're talking about 500 cells per group, and it just translates into really, really large amounts of quantitation. So this quantitation is, first of all, very time consuming. It's labor intensive, and depending on who the person is who's performing the analysis, there's going to be a large amount of error and bias that is associated with the data quantification. So we wanted to devise a method that would determine the area of the Golgi and the cell, or area of the Golgi and the nucleus, by assigning some specific biological constraints. And we thought that this would help us eliminate the current challenges that I was facing in the analysis. And as Kayal mentioned at the beginning of the talk, so this was the problem statement that I brought to the table during the summer workshop that was organized by the Center for Analysis of Biological Data. So at the summer workshop, we had about a week where we teamed up with participants from other universities who were from other streams other than biology, and they gave us a lot of good ideas and good strategies to overcome the challenges for this project that I was working on. And at the end of that 1 week, we were encouraged by the center to apply for an interdisciplinary research grant that would allow us to continue our collaboration over the year and see if we can come up with a novel method to overcome these challenges. So once we got the grant, all of us we got together a few times and worked on various strategies and various algorithms to overcome these problems. So the final rationale behind the approach we chose was going to be based on MATLAB. We had tried many other approaches, but we found that the MATLAB-based approach worked best for us, particularly for this problem statement. And we decided to center the nucleus as a frame of reference when it came to identifying individual cells and also the Golgi that is associated with the cells. So we have been working on this for over a year, and over time we had another faculty member from the Department of Statistics, Dr. Walid Sharabati. He works at Purdue in the statistics department. So he also joined the team, and this team was headed by Dr. Claudio Aguilar and Dr. Mark Ward, who is from statistics, and Dr. Walid joined us and guided the students in the team in creating a very objective method towards quantitatively assessing the biological phenomena that I was studying. So I'm just going to briefly talk through the stage at which our current tool or methodology is at. So as I mentioned, it's based on MATLAB and uses the nucleus as a frame of reference. So before I go into the details of the outputs that you're seeing on screen, one of the major challenges that a user would face when it comes to analysis like this is there are existing softwares that could help in determining areas of these structures. But one of the problems associated with that is these softwares, when they apply, let's say, a threshold or a piece of code to identify individual structures, they either underestimate the area that we are interested in or overestimate the area that needs to be measured. So one of the biggest challenges with the pre-existing automated tools that we have is that it lacks in accuracy, which a human user can make up for. But the speed at which a software or a tool can do this analysis is definitely much, much better than what a human user can do. So on the left, you see an example or an input image. These are fluorescent images of patient cells. So these are cells from Lowe syndrome patients. And as I mentioned, they have been fluorescently probed in red with a marker for the Golgi, and in blue you can see the nucleus associated with each cell. So the first piece of analysis that we're doing is we're trying to establish a threshold for this color image. So why we need to do that is we need to be able to identify discrete parts of all the structures that we see in this image without taking in any area from the background or eliminating what should actually belong to a discrete particle. So we use derivatives where we try to track the rate of change of color over a given distance and then use that measurement to attach a particular threshold to this input image here. And once you choose a particular threshold, it's going to be creating discrete structures which will later be analyzed for their area and things of that sort. But the next important thing, which is the only step where we have human intervention in this tool, is we want to let the code know what are the cells that we want to take into this analysis and what are the cells that we would want to eliminate from the analysis. So if I was doing analysis on this image manually, obviously I would be eliminating cells that are in the corner that probably aren't quite as clearly visible as cells right in the middle of the image. Or I would also eliminate cells that seem to be lying on top of each other. So these are biological constraints that we would want to input into the code or into this tool in order to ensure the analysis is done correctly. So the second step would just be marking out the cells that you want to eliminate from the study, and also selecting the cells that you want to take into the study. And once that is done, you get this image on the far right bottom. So as you can see, I have eliminated cells at the corner of the image that didn't quite fall into the frame. And we also now have cells that are visible in the whole, and they do not seem to have any overlap amongst themselves. So finally, what the code is doing is it's locating the centroids of the various nuclei that we want to analyze. So the red spots on this top right panel here are the centroids that are located in an iterative process by this code. And once it locates the centroid, we then get the code to quickly calculate the area of these distinct particles using a clustering algorithm. So we have the centroid for a given cluster. It's very easy to determine the area for that particular cluster. And we perform the same analysis on the Golgi part of the image. So they are separated out into channels initially. I'm sorry, I forgot to mention that. So first, the analysis is performed on the channel that carries only the nuclei, and then a similar analysis is done on the channel that carries only the Golgi, and then we have the centroids associated with those clusters of Golgi, and then we also have the areas. And finally, what the code is then designed to do is match the nucleus with the Golgi. So based on the positions of the centroids, the nucleus and the corresponding Golgi are matched, and with the areas in hand, it provides us with an output that is actually the ratio between the area of the Golgi to the area of the nucleus, which is ultimately the information that is biologically relevant, and that is the information we seek from this analysis. So this is just a sample output from both healthy cells. The top panel shows you a fluorescent image of healthy cells that are again stained for the Golgi and the nucleus, and the bottom shows patient cells that are stained again for the Golgi and the nucleus. So the output that you see here are ratios of the areas, as I mentioned, and we want the output to be given to us in a form where it gives us the ratio for each cell in the image and not for the overall image. So for our healthy cell analysis, let's say we collected this information from 10 different cells. The software or the code is able to give us that information for those 10 individual cells, and it does the same for our patient cells as well. And finally, we can perform any kind of statistical analysis that we want on it, such as median or determining standard deviation, to see if the data that we get from this calculation is going to be statistically significant. So in conclusion, from where we began back at the summer workshop, we now have a tool that can automatically determine discrete components such as the nucleus from a biological image, and also Golgi structures from a biological image, which are two-dimensional from fluorescent microscopy images. And we are now also able to quantify the areas of the nucleus and the Golgi and match them with one another and calculate the ratios of the two areas as well. So to make this more biologically valid, one of the things that we are now working on is to perform the same analysis but in a three-dimensional scenario. So we want to ultimately determine volumes of these compartments of the cell that I'm interested in to see the change in the morphology of the Golgi and compare it against the nucleus in the cell. So we are currently working on how to take this from a two-dimensional scale to a three-dimensional scale. So in conclusion, I would really like to thank Dr. Claudio Aguilar and Dr. Mark Ward, who initiated the summer workshop where we began this biological data analysis study, and this was where our project began. And we would also like to thank the center for giving us this opportunity to apply for a grant, particularly Brent Ladd, who really encouraged us to apply for this interdisciplinary grant, and now also for giving us this opportunity to talk to everybody about the work we've done and also our collaborator, Dr. Walid Sharbati from Department of Statistics here at Purdue, and also the students on this center-sponsored interdisciplinary grant, Priya from Texas A&M, Sundar from Johns Hopkins, and Yukong here at Purdue, and our funding sources for helping us move forward with this project and also give us this opportunity today.